Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to talk about applications of IR spectroscopy in organic and inorganic compounds. So, identification of substances. To compare spectrums, no two sample will have identical IR spectrum. Criteria: samples and reference must be tested in identical conditions like physical state, temperature, solvent. Disadvantage: enantiomers cannot be distinguished. So the fingerprint reasons small differences in structure and constitutions of molecules results in significant changes in the peaks in these regions. Hence, this region helps to identify an unknown compound. You can see the percent transmittance, wavelength, CH stretching, CH bending, CH stretching, CH bending. Computer search system. Newer IR instruments offer computer search system to identify compounds from stored infrared spectral data. The position and magnitude of peak in the spectrum is compared with profiles of the pure compound stored. Computer then match profiles similar to that of the analyte and result is displayed. So you can see the benzenes and your compound is unknown, and then you compare it. Another important applications: determinations of molecular structure used along with other spectroscopic techniques. Identification is done based on position of absorption band in the spectrum. Absence of band of particular group indicates absence of that group in the compound. Third application is studying the progress of reactions. Observing the rate of disappearance of characteristic absorption band in reactants or the rate of increasing absorption band in the products of a particular product. So OH and carbon double bond oxygens. Now friend, detection of impurities determined by comparing sample spectrum with the spectrum of pure reference compound. For example, ketone impurity in alcohols. Detection is favored when impurity possesses a strong band in IR regions where the main substance do not possess a band. Isomerism in organic chemistry. Geometrical isomerism, trans isomers give a simpler spectrum than cis due to symmetry. Conformers, rotational isomers, identify with the help of high resolution IR spectrometer. So, ethanol normal OH 36, 136, and weak band 36, 122. So you can see the NT formula of horse and skew. Tautomerism, evidence or existence of two or more chemical compounds capable of interconversions, usually by exchanging a hydrogen atom between the two atoms. To see the thiocarboxylic acid. Functional group isomerism. Isomerism shown by compounds having the same molecular formula but different functional groups. You can see the diethyl ether and ethanol. Application of IR spectroscopy to inorganic complexes. Difficulty is high mode of vibrations since they contain more than 5 atoms. Hence minimum 10 mode of vibrations. Lower symmetry of complexes due to formation of ligand and polynuclear complexes. Formation of chelates. Chelates, large number of new bands appear. So assumptions not always true. Complex formations only affect the vibrations of the ligand slide. Vibrations do not undergo coupling with other vibrations of another ligands. When ligand is coordinated with another atoms, it will not change the symmetry of the ligands. So, geometrical isomerism by pyridyl cobalt trichloride. 2-isomer trans isomers has more symmetry than cis isomer. Hence, complex spectrum is expected for cis isomer. So, shape of symmetry of a molecule. Nitrogen dioxide. If linear, only 2 bands should be present. If bent, 3 bands should be present. Actual spectrum shows 3 peaks at 750, 1323 and 1616. Inverse centimeter. Similarly, II spectrum was used to determine the structure of XCF2, XCF4, XCF6, linear, square, planar, and octahedral system. Identification of functional group. Due to presence of functional group regions, you can see presence of water in sample. If lattice water is present, spectrum will contain 3 characteristic bands at 3600 to 3200, 600, 1650 centimeter inverse, and 600 to 300 centimeter. Measurement of paints and varnishes. Measured by reflectance analysis. Measure IR absorbance of paint on appliance of automobile without destroying the surface. Make a year of car can be determined from IR spectral analysis. Examination of old painting and artifacts. Help to determine the fake masterpieces. Varnish and paint from old item, statue, canvas are analyzed by IR spectroscopy. Presence of new paint traces implies the masterpiece. So, in industries, determine the impurities in raw materials. For quality control check to determine the percentage of required product. Identification of material made in industrial research lab or material of competitors. So, analysis of petroleum, hydrocarbons, oil and grease contents. These contain CH bond, absorptions at 3100 to 2700 centimeter. Freons, fluorocarbon do not contain CH bond. Thus, quantity of hydrocarbons, oil and grease in freons is determined by measuring CH absorptions at. So, quantitative analysis of multi-component mixture of sulfur-oxygen anion by FTIR spectroscopy. FTIR help to determine sulfur-oxygen anions in aqueous solution. SO stretching band at this range. FTIR uses water resistant cell, have short and reproducible effect path length. Characterization of heterogeneous catalyst by diffuse and reflectance spectroscopy. Diffuse reflectance spectroscopy helps to determine the nature of molecules attached to catalyst surface. Characterization of olefin polymerizations, catalysis with silica gel, different type of silicon OH bonds are determined. So, analysis of multilayer polymeric film using FTIR spectroscopy. Determine the identification of polymer material in multilayer level. FTIR help in quick characterization. Other application like determination of unknown contaminant in industry using FTIR, determination of cell wall of mutant and wild type of plant varies using FTIR, biomedical studies of human hair to identify disease status, recent report, identify, order and taste components of food, determine the atmospheric pollutant from the atmospheric itself. So, quantitative analysis. Quantitative analysis based on the determination of one of the functional group. Concern of hexanol in hexane hexagonal mixtures. So, you can apply Beer-Lambert's law. Method for determining A and concentration C. Cell in, cell out method, standard calibration curve method. Baseline method, selections of suitable absorptions, band, P0 and PR measures. So, baseline method like this. What is the advantage? Common possible error are eliminated. Same cell is used for all determinations. All measurements are done on points defined by the spectrum, means no dependence on lambda intensity, and eliminate changes in instrument sensitivity and source intensity. So using KBR pellet disc, uniform pellet of similar weight and thickness, known weights of KBR, known weights of test, calibrations curve plotted, discs are weight and thickness measured, using internal standard potassium thiocyanate, dried ground with KBR to make concentrated 0.2% by weight of thiocyanate, calibration curve plotted, ratio of thiocyanate absorptions to a closed band test is plotted versus concentration. So friends, I hope this video you like it, and please subscribe my channels. Also share in your friend circle through WhatsApp. So if you like this video, then subscribe my channel, and thanks for watching.